my name is Miaoqing. I'm from MIT. The talk today I'm going to give is that stochastic cytokine expression induced mixed T cell states. So this diagram shows you the current model of gene regulatory network governing differentiation of T helper cells into Th1 versus Th2 lineage. So the Th1 lineage help our body to defend against intracellular pathogens such as viruses and mediates the killing of the infected cells, whereas the Th2 deals with extracellular pathogens such as a worm infection by eliminating the pathogen. It is very important to achieve an intricate balance between Th1 and the Th2 response upon infection. And excessive response of either Th1 or Th2 can be harmful because it can induce autoimmune disease or allergies. So in this diagram, the players for Th1 pathway is color-coded in red, and that for Th2 is in green. The master transcription factor for Th1 pathway is Tbat, coded by the gene Tbox21. And that for Th2 pathway is GATA3. So both Tbat and the GATA3 can induce their own expression either directly or through other transcription factors. And Tbat can upregulate the expression of interferon gamma, which is the Th1 cytokine. And once interferon gamma is secreted to the media, it can trigger signaling cascade that induces the expression of Tbat. And a similar relationship exists between GATA3 and IL-4. And there is mutual inhibition between Tbat and GATA3 and the inhibition by Tbat on IL-4 and vice versa. So by looking at this current model of signaling diagram, it is predicted that the expression of Tbat and GATA3 must be mutually exclusive in individual cells. And also, the expression level between Tbat and interferon gamma must exhibit positive correlation, and that between Tbat and IL-4 must exhibit negative correlation. So to test these predictions, we examine the differentiation of T helper cells in the cell culture. By isolating naive T helper cells from the mice spleen and lymph nodes and culture them on Petri dish, which is coded by antibodies against cell surface receptors such as CD3 and CD28. So the functions of these antibodies is to induce clustering of these cell receptors, thus activating them, leading to activation of these cells. And this whole differentiation process usually takes a couple of weeks. But in our analysis, we focused on the early time points because we want to see how the cells make a decision. And distinguished from existing studies, in our design of the experiments, we did not bias cell fate by adding extra exogenous cytokines, and we measured the gene expression in single cells instead of at the cell population level. And we also focused mainly on the early differentiation time points. And then the method we are going to be using for the quantification is single molecule FISH, developed in our lab. So in this methodology, a large number of 20-mer DNA probes is designed, and each is coupled to a fluorophore to tie across a single target mRNA. So the co-localization of a large number of fluorophore to a single transcript will enable you to see a very strong diffraction-limited signal under epifluorescence. So basically, you can see individual transcript as an individual spot under the microscope. So with this methodology that gives us single molecule resolution, we see very striking results as shown here. So these are the cells at 24 hours after activation. In red is Tbat, and in green is GATA3. So contrary to what the current model predicts about mutual exclusion between Tbox21 and GATA3, very strikingly that we see high-level co-expression of Tbox and GATA3 in individual cells. And distinguished from the low-level co-expression as in the priming model that has been described previously, the co-expression we have observed here is at very high levels, which is comparable to terminally differentiated cells. So this shows you a picture of terminally differentiated Th2 cells, and the GATA3 is in green. That you can see that even at 24 hours, the expression level of GATA3 is pretty comparable. And to quantify our data, we did a scatter plot. So you can see that at 0 hours, the naive cells basically do not express any GATA3 or Tbox21 transcripts. And as time increases, we see this high-level expression, co-expression of Tbox21 and the GATA3. And then if we see the mean level, we basically see the general trend of increase. And again, to compare the expression level with fully differentiated Th2 cells, we can see that at 48 hours, we get a mean number at about 80, which is very comparable to the experiment here. So having examined the expression level of the transcription factors, then we questioned what's the expression level of the cytokines in the individual cells. And we performed the experiments. And again, we see very striking results. So in these images, color-coded in red are the cytokines. Interferon gamma in this image and IL-4 in this image. And in the green are the transcription factors, Tbox21 here and the GATA3 here. So from these images, first you can see that again. We can see that the transcription factors in green are ubiquitously expressed in many cells. But strikingly, the expression of cytokines such as interferon gamma is essentially off in most of the cells. And it's expressed only in a rare cell population, for example, in this cell. And once it's expressed, it seems to be at a very high level. And below are the histone diagram, and we see similar results for IL-4 expressed in a rare cell population. Then we examined the correlation between the expression level between the transcription factors and the cytokine. So I just mentioned that the current model would predict that there should be positive correlation between interferon gamma and the Tbox21, and between IL-4 and the GATA3. However, by looking at this data, you can see the positive correlation is rather weak. And the current model would predict there should be negative correlation between interferon gamma transcripts and GATA3 and between IL-4 and Tbox21. But again, we didn't observe any negative correlation. So then we questioned, can we manipulate the cells by manipulating the amount of cytokine available to the cells? And yes, we could. The way to do that is to add neutralizing antibody that would sequester cytokine away from the cells. And we performed the experiments. And we have observed that by adding antibodies, neutralizing antibody to sequester away the cytokine, then the expression level of the transcription factors are downregulated. So as shown here, if you add antibodies against interferon gamma, the expression of Tbox21 is downregulated, while the expression level of GATA3 remains unchanged. And the reverse result is true when you add IL-4 antibodies. So to facilitate the interpretation of our data, we converted this density, scatter plot to a phase angle plot where we use the parameters r and theta instead, which r is the distance from the origin and theta is the angle. So a small theta would mean that the cell is lying towards the x-axis and therefore Th1-like. And the large theta will indicate that the cell is Th2-like. So extracting the information from the scatter plot, then we get the information about theta, and we also exclude cells which have r smaller than 10 because these cells will have a noisy value of theta. Then we can convert this scatter plot to a density plot of theta. So from this scatter plot, you can see that as T cell can be in a state anywhere between the x-axis and the y-axis, and when you look at the angle theta, it forms a uniform distribution. from 0 degrees to pi over 2, which is 90 degrees. So with that, we can convert our data all using this phase angle density plot. So if you add interferon gamma antibody, the cells will populate in the large angle beams. It exhibit a Th2-like behavior. And if you add IL-4 antibody, as the antibody concentration increases, the cells will become more Th1-like. So then we came back to revisit the current model governing T-cell differentiation. So from our data, we have seen that there is no positive correlation between Tbat and interferon gamma, so we grade this activation arrow. And we didn't see any negative correlation between Tbat and IL-4, so we also grade this inhibition arrow. So then we got rid of the arrows, the regulatory arrows here, at least during early differentiation phase. In addition, we also have seen co-expression of Tbat and GATA3 at high levels. That would mean that the signaling strength from the cytokine must be very strong, that it would overshadow the mutual inhibition between the transcription factors. And the T cells can essentially be depicted by this model. So here, in the oval are the transcription factors, and the triangles are the cytokine. So a rare cell population can stochastically turn on the expression of the cytokine. And the cytokine, after being secreted to the media, it will induce the cell to express transcription factors at very high level. So to test our model, we can try to remove both cytokines. That will leave us with this only intracellular network. And from this network, we would predict that we should see mutual inhibition of Tbat and GATA3 in this case. And then we performed the experiment. And indeed, from this scatter plot, you can see now the data points are more concentrated along the axis. And if you look at the phase angle density plot, instead of seeing a uniform distribution, now you see a U-shaped distribution with densities concentrating near 0 degree and 90 degree. So with that, I'd like to conclude my presentation. And to summarize our result, we have observed high-level co-expression of antagonistic transcription factors in single T helper cells during its differentiation. And the cytokines are produced by a rare subpopulation, and their expression is not correlated with Tbat or GATA3 levels. And the role of the cytokine, namely interferon gamma and IL-4, is to upregulate the expression of the transcription factors. And the strength of cytokine signaling must overrule the mutual inhibition between Tbat and GATA3 within the cell. And with that, I'd like to thank my advisor, Alexander van Woudenarden, and thank my lab mates. And I would also like to thank my collaborators in Cambridge University, Sarah and Daniel, and those in the Whitehead Institute, Huang Ming, Harvey, Stephanie, and Heather. And I'd like to thank the RECOMB conference for giving me this precious opportunity to present here, and I thank you for your attention.
Wonderful talk. We have time for one question. I encourage you to move quickly to the mic so we can make up for some lost time. Okay, here we go.
Well, I guess one of the statements that you had is that you can actually modulate whether or not it's a Th1 versus Th2 response by sort of manipulating the environment. I was wondering if that's actually ever been used clinically, like say in leprosy where you have this sort of balance between Th1 versus Th2. Has it been shown, or in any other sort of disease processes, that by giving interferon gamma or by sort of modulating IL-4 that you can actually benefit the disease itself?
So your question is whether the use of neutralizing antibody has been used before.
Or not even neutralizing antibody, but the sort of opposite of that, which is to overwhelm the system with, say, the interferon gamma or the IL-4 to sort of switch over from, say, what is, disadvantageously being a Th1 response, but should be a Th2 response.
Yeah, that's a good question. So we did perform the experiments by adding exogenously at a high concentration with cytokines into the cell culture media. And what we have seen is that because that we still see the high-level co-expression that are comparable to the results shown here. So I think what basically is happening here is that the cytokine level that is produced by this subpopulation is already at a saturating amount. So even by adding more exogenous cytokines, you don't really change the expression pattern. You still see that high-level co-expression.
Right, let's thank Miaoqing again.